Thanks everybody for joining us for the resumption of our Oregonian Science webinar series hosted by the Great Lakes Fishery Commission, for which we're very grateful. Renee, you want to announce our next webinar people can sign up for?
Yes. Well, first of all, thank you everyone for joining today. We know it's been a bit of a long hiatus. So we've got Dan presenting today, and then in January 2026, we have Kristen Behrens presenting genomic insights into population structure and sex determination in Great Lakes Cisco's. And I'm going to drop the link in the chat for registration for that as well. And then the following Friday, January 16th, we are going to have the long-awaited Randy Brown presentation. So if you've already signed up for that, that link will stay the same. We are finalizing a time for him since he's in a bit of a time zone, 4 hours away from us. So we will resend that invitation when we get the final time. But it's great to have you all back today.
Yeah, good looking turnout so far. And a plug for Kristen. She's been working on some low coverage whole genome sequencing, some really hot off the press information that she'll share next month. So I'm going to introduce Dan, and Dan can start sharing his screen. This is Dr. Dan Isermann. Dan got his bachelor's in science at Southern Illinois University. Went to Tennessee Tech University to get his master's, working with Phil Batoli, a great guy that I interacted with some as well. Then moved on to South Dakota State, where he got his PhD with the late but excellent Dave Willis. Dan is also with USGS, as well as he's in the Cooperative Fishery Research Unit at the University of Wisconsin, Stevens Point, and the College of Natural Resources. He supervises a wide array of research on economically and culturally important fish populations in the states of Wisconsin and Michigan. And a significant focus of that is the whitefish. And you'll hear more about that today, obviously. His research requires a lot of coordination with state and tribal management agencies. Also works really well with commercial fishers and guides. Throughout his career, he's advised more than 30 graduate students that work as fishery biologists throughout the country. He's also an editor with the North American Journal of Fisheries Management, which is quite an honor. Other honors include a member of the AFS Fishery Management Section's Hall of Excellence, and he will be a 2026 inductee into the Freshwater Fishing Hall of Fame. I think it's more for his research contributions. That's what it says, but maybe also for his fishing. I don't know. He'll have to tell us. No, okay. Yeah. For his research contributions. So a lot of great honors coming Dan's way. So we're really tickled that he accepted our invitation to come here and talk about that lake whitefish stock, especially with what's been happening in the tributaries in southern Green Bay. So you want to flip it, like switch presentation mode there, Dan? So we're kind of also seeing your presentation view or your presenter view.
Okay.
It worked before, we know. I think there's an option just to swap. Here we go. That's it.
Got it?
Got it. So I forgot to mention that Dan's talk is about 40 minutes or so, but we really do hope that you all will stay on, as we often do, and have a really nice discussion, question and answer session. And Dan's willing and excited to chat with you all. So with that, I cede the floor to you, Dan. Thanks.
All right, I appreciate it. It's definitely whitefish weather here in Stevens Point, Wisconsin. So getting some snow and some sub-zero weather, and people were ice fishing on a couple spots this morning on my drive in. Again, maybe not smart people, but people were out fishing on the ice. I'm going to really focus. We've been doing a lot with whitefish, mostly in and around Green Bay, but I really wanted to focus in on the whitefish that are spawning in the tributaries to southern Green Bay, just because that's kind of I'll say a unique behavior currently in Lake Michigan. And I really want to first recognize there's a whole bunch of folks that made this all possible. my co-authors. I'd especially like to thank Dan Demkowski, who works with me here at the co-op unit, for helping me get this together. Our partners with Wisconsin and Michigan DNR that help us with tagging and a lot of other things. And then really two major contributors. The funding for most of the work I'm going to talk about today has come from the Great Lakes Fish and Wildlife Restoration Act, and that comes through the U.S. Fish and Wildlife Service. And then also we've had tremendous support from our folks up at Hammond Bay with GLaDOS in terms of analyses, help with infrastructure, lift and receivers, you name it. And also just with the data portal, like Nancy Nate has been incredible helping us wade through this. All right. I want to just talk a little bit about our program. Most of you are probably familiar with the co-op units, but we're really focused on conducting applied research. So we're USGS employees stationed at these universities throughout North America, providing technical assistance and then also training graduate students. And I think this work with whitefish is a good example of all of these things. I didn't know how diverse of a group of folks we might have today, so I'm going to provide a little bit of background information. This is the species that we're going to be talking about today, lake whitefish. Um, and I also really wanted to orient people with this part of the world. Instead of a north arrow, we're going to use the Green Bay Packers symbol at the bottom of the map here to orient ourselves. So that's Green Bay, Lambeau Field, the south end of Green Bay. Um, Green Bay is falls both within the states of Wisconsin and Michigan, and the Menominee River there serves as that border between Wisconsin and the UP. We're going to talk about some of these locations quite a bit, like Big Bay to Knock, North and Moonlight Bays. We're gonna talk about that. The one thing I really want you to remember is that big CI in the middle of the Green Bay there is Chambers Island, and that's the delineation we're going to use for North and South Green Bay. And then you see the Door Peninsula there. You're also gonna hear me say things like Bayside and Lakeside, and that's what I mean when I use that terminology. Most of you, or a lot of you, are probably already familiar with the fact that lake whitefish are one of the most commercially important fish species in the Great Lakes. Also very important to many tribal communities. And then we're going to talk a little bit about importance to some recreational fisheries. And then again, just to generalize how we manage harvest of these species in the Great Lakes, we use these management zones, catch and age models for individual zones or cluster zones. We also know that lake whitefish recruitment is supported by multiple stocks within Lake Michigan, and we'll talk a little bit more about that in a minute. And we definitely have mixed stock fisheries in some of these zones. And the important thing to remember here is that the fish don't know where these lines are, and they're going to cross them when it's suitable for them. And so we're going to go back just a little background information about, again, we're going to focus on Green Bay here, but previous work, and this would date back even to Mark Ebner's work early on with tagging. We kind of had this conceptual model, and genetics confirmed it to some degree, that we have these different stocks of fish that are contributing to the Lake Michigan fishery. And the one thing that is really interesting is Justin, when he did this work, they went and sampled fish from all of these locations to figure out that these stocks existed. But no fish were collected from southern Green Bay or the tributaries. And this was the case for many, many previous studies, and actually many studies since then. And the reason was that this just really wasn't that big of a thing. Like lake whitefish in southern Green Bay were not necessarily on the radar to any great extent. But a lot has changed over the last several decades, and the big thing, or one of the most interesting things to me, has been the resurgence of these tributary spawning groups in southern Green Bay. And that's primarily the Fox and Menominee rivers. That's what we're going to talk about today, but there are also smaller runs in the Peshtigo and the Oconto rivers. And then some other things have also happened that kind of generally coincided with the tributary spawning. We see a pretty good increase in lake whitefish abundance in southern Green Bay. And at the same time, or at least on a similar timeline, we're starting to see recruitment decline in some of these other locations like Big Bay to Knock and the lakeside of the Door. And there's a shift in commercial effort, reflecting the shift in fish abundance from the lakeside to bayside. And then this winter ice fishery kind of emerges at about the same time that, we really notice the tributary spawning. This happens in a lot of different locations, and harvest in some years can exceed 100,000 fish. It's really contingent on whether we get ice or not, and that has been not consistent over the last several years. But maybe you can tell here from the bottom picture, it's so easy that even like Ted Tresca and I can catch a whitefish out on the ice. That isn't true every day. It can be pretty tough on some days. So there's a lot of speculation that kind of arises out of all this. And one of them is just like, where did these tributary spawners come from? How did it start? And maybe it was due to habitat restoration, leading to cleaner rivers and better habitat. And then fish from these other spawning locations recolonize the river. And at the time, people were really thinking that maybe these are fish from like Big Bay to Knock or the lakeside of the Door Peninsula. So they'd be coming from pretty far away. Or maybe there were always some fish doing it, but they just kind of went undetected. And then some other things, this sort of temporal alignment suggests that, well, maybe these tributary spawning fish are really responsible for all these whitefish in southern Green Bay, and that these fish are supporting this winter ice fishery. And maybe these fish provide a source of recruitment for other places, like potentially there's some spillover. And even though things aren't going well elsewhere, maybe some of these fish are moving to these other locations. So the first phase of our research was really focused on these questions. And again, we tagged in several other places, but I'm going to focus on the Fox and the Menominee. Where we wanted to know, where did they go after they spawn? And remember, they really hadn't been included in previous evaluations. Are they loyal to these spawning locations? What do their genetics look like? Could they possibly contribute to fisheries in other places? And then to what extent? Was another question that we had. So our approach to this, and it was to combine conventional tagging with more advanced genetic techniques like genomics and SNPs with acoustic telemetry to address these questions. And a lot of this is summarized in Lisa's paper here that's in North American Journal of Fish Management. I just have a snip here if you want to learn more about it. You could go to the paper. I'm going to generalize quite a bit. So the first thing that we needed to do is figure out if whitefish would live through this process of being tagged and how best to sort of knock them out for the surgery process. So we did these surgery trials inside Menominee Dam. We used the lake sturgeon transfer tanks that are used there. They're big, round, and provide a constant flow of water, and that's going to be really important. We're going to talk about that a little bit more. And then we compared two different methods of sort of, again, like knocking the fish out or immobilizing them during surgery, and that included the TENS unit up at the top there, and then also aqueous. And this was really successful. We had really high survival. Maybe one or two fish died, and I think they might have actually not been, they were sort of our treatment or control fish, I should say. The one issue with the aqueous is that at really cold water, the recovery times were really, really long. So out in the field, we wanted to go with the TENS unit, and that's what we did. So in 2017, we worked with the Wisconsin DNR and we tagged 101 fish in the Fox River and 100 fish in the Menominee River, and they were caught via electrofishing. And then we held them in these air-ready tanks both before and after tagging. And what I want to note is that this is not a big round tank with a constant flow of water moving through it. And we think that might have contributed to some of the results we're going to see here in a minute. The fish looked healthy and they looked happy, but they didn't have a lot of space and they weren't being pushed to swim. We used V13 transmitters. The yellow thing at the top is actually a temp logger that went in the fish along with the transmitter. This was a little bit before the built-in temp depth sensors were available in the tags, or at least it could have been cost prohibitive. I can't remember. They got an orange loop tag that indicated a $100 reward. And then at the same time, the Wisconsin DNR and other folks were loop tagging fish with green non-reward tags. So occasionally we're going to refer to those loop tag recoveries. And this is what our receiver array looked like at the time. We were mostly interested in transitions between these zones. So again, I'll hold my mouse over it. This the receiver lines here in the middle of Green Bay are that Chambers Island that we're going to use to denote North and South Green Bay. And then the receiver lines up here by Washington Island would tell us whether a fish went out into Lake Michigan. And so this is what we learned from those fish. I just kind of simplified this. This is an old heat map we had. Where you see the brighter red colors or like shades of pink, more fish have been detected on a receiver or a receiver in a line. Where you see the X's, no fish were detected. And so here's our Menominee River fish. Remember, we tagged 100 of them, and we ended up keeping 60 fish in the analysis. And so our expectations were, you tag 100 fish just due to normal mortality. Like a year later, maybe you're going to have 70 to 75 of those left. And so potentially some tag related mortality here, but at least a decent sample size. And what we saw is that about a third of these fish spent some time in northern Green Bay. And a few fish went out into Lake Michigan, but not very many. The realities of it are the ones that did go north often didn't spend a whole lot of time north of Chambers Island, and most of them stayed south of Chambers. So they were in southern Green Bay and not going up to Big Bay to knock or going out into Lake Michigan. And our loop tag recovery showed a very similar pattern. The Fox River was a little more interesting because only about 16 of these fish yielded us usable data. So we definitely had some mortality issues. But what we saw from those fish, and again from the loop tags, was that these fish really do not go north of Chambers Island. They are a southern Green Bay group of fish that are spawning in the Fox River. And then just fidelity wise, just interest, you can see here, they're pretty loyal. Most of the fish return to the same river to spawn in subsequent years. They didn't stray between the two rivers. The only thing we saw is in the Menominee, there were definitely fish that were alive and out in the bay in like the middle year of the assessment, so they skipped coming into the river. And whether that means they skipped spawning or they spawned out in the bay, we don't know the answer to it. We weren't happy with the outcome, the mortality levels that we saw, especially for the Fox River. So we did this again. And this time we used the big round tanks and force water through them. And I'm just going to play this little video so you can kind of see. This is a tank we used to move them back out into the bay. And we've had like much better survival. We're much closer always to that like expectation of how many would die from just natural mortality. And then we also did some comparisons on survival again with the TENS unit versus the Aqua-S, just to see how well they did out in the wild when we use those two methods. Whoops. All right, so these are sort of the results from that phase 2, and this for the Menominee River is pooled. So it's all the tags, both from that first tagging event and then the second tagging event, plus the loop tags. Broken out by season. When you look over at the map, the bigger the circle, the more detections that we had within that region. And so you can see that The bigger circles tend to be in southern Green Bay. And again, like not a ton of fish are going up to the Bays de Noc up on the north end of that map or out into Lake Michigan. So pretty consistent with what we saw from the first round of acoustic tagging. Well, we had a pretty big change in operations before we tagged the Fox River fish, and that was we switched to the grid. And so this is the grid that we currently have out in Green Bay. They're spaced at, I always get this wrong, 8 kilometers, I believe, something along those lines. Dan can correct me. It's been a while. But a very different array for us to be able to really get a better idea of where those fish might be, even if they cross into northern Green Bay. And at this point, we also started to get significant help from the Green Bay Fish and Wildlife Conservation Office in tending these receivers because it got a little bit trickier. And then we also largely converted over to acoustic releases instead of using the snag line. So some pretty big changes in our approach to things. So to kind of like keep along the same analysis approach that we've shown in the other slides, Dan actually grouped the receivers into these zones so we could see like how many Fox River fish like actually made it north of Chambers Island. So most of them still stay south of Chambers. So like 3% of them, 13% of them might have made it to Chambers Island, but only about 3% of them end up in that northern portion of Green Bay. So this just sort of reaffirms what we saw in the first round of tagging, that these Fox River fish are really staying south and probably not contributing a whole lot elsewhere. And then the genetics were also really interesting. So all of the fish that we tagged in these rivers get a fin clip, and we have a genetics lab here on campus that was run by Jared Hamola while he was here. And what we learned is that there are two stocks of fish in these rivers in southern Green Bay, and essentially we're calling these northern and southern stocks. And the one key point is that we used to think Big Bay de Noc and the lake side of the door were a separate stock, but the new analyses indicate they are not. They're not separate, and that makes a lot more sense based on what we know about their movements. When we look at the fish spawning in the Menominee River, they're a mix of pure northern and southern fish, but also a fair number of what looks like hybrids. The Fox River fish are also a mix, but a lot more of them are pure southern fish, is how I would characterize it. And the one thing that we've been able to tease out now, and this is in a forthcoming paper, is that telemetry suggests both of these stocks are spawning at the same time in the Menominee River. So there isn't some like distinct run between the two. All right, I just want to kind of summarize our thoughts on that first round of stuff, so to speak. And what we were thinking is, based on our findings, that the Fox River fish are probably pretty unlikely to contribute to fisheries anywhere else but southern Green Bay, and that the Menominee River could maybe contribute in other places, but there's not really very many fish moving north. But we'll come back to that not very many fish moving north point here in a minute. And then we gained some more information, things, more research is going on, more catch at age estimates are coming out of the models. There's a lot of fish in Green Bay, like based on assessment and modeling. I mean, millions of fish. But the larval output that's coming out of the rivers doesn't really seem to be sufficient to support this many fish. And so our colleagues at UW Green Bay had done some work and tried to estimate larval output. You're talking somewhere maybe hundreds of thousands to low millions of larvae leaving the rivers. And then our grad student, Jordan Everson, was doing a ton of larval sampling around the bay. And we were just seeing really large concentrations of larvae in places that were pretty far away from these rivers. And what it indicated to us maybe is that we do have some reef spawning in southern Green Bay. And there were other people that had thought this previously, and there have been some efforts to identify, where is this happening. But we were starting to really wonder, how important are the tributaries actually to to the fisheries in southern Green Bay. And so that led us to phase 3 of our research. And this is kind of the opposite of what we were doing before. Now we're going to go out into the fishery and tag fish and then see where they go to spawn. And that was our goal, was 150 fish from each fishery, both the commercial and the recreational ice fishing, over several years. We've completed the commercial catch tagging, but we still need to tag 80 fish on the ice. And this is just one of our fish here to the right. You saw swim off there. So I think we're gonna get ice again this winter, so we should be able to knock out these 80 fish. So I want to just show our very preliminary results here, and this is just from the first cohort of commercially caught fish that we did in 2022. And those fish were caught over here in Sturgeon Bay. I'm trying to highlight it with I should have put something on the map here. I apologize, but they're over here in this region. And this is where they were in the next spawning period. November 12th would be right around the peak of the lake whitefish spawning window. And the hotter the color, the more fish we detected. And there's receivers in the Fox and Menominee rivers, and you'll notice they're not lit up at all. From what we can tell, only like 3 fish might have gone into the tributaries out of that original tagging group. I don't know. We tagged 75 fish, and I think we're showing data for like maybe 50 some of those fish here. What we do see is maybe like a loose aggregation out on these shoals that are out in front of the tributaries. And this is really preliminary, but if this holds true, that would suggest that the tributary fish are probably not very important to that fishery, which is really interesting because we know a ton of the Menominee River fish spend a lot of their time over by Sturgeon Bay where we tag fish. So a lot of Menominee fish go there, but they may not actually be like that important of a source of fish. Again, if this holds true, it's still really preliminary. And then we were still thinking about the effects this might have on other places. So what about northern Green Bay? Again, like not a lot of the Menominee River fish are going north, but a little bit might help a lot in places that might have declining recruitment, is the way that we were thinking about this. So that's kind of part of our ongoing research that we're doing now up in Big Bay to Knock. We're kind of repeating again, where do the fish spawning in Big Bay to Knock go? But what's new about what we're doing is we're tagging fish outside of that spawning window and trying to see where they go to spawn. Are there any Menominee River fish in these nets, or is it just like local Big Bay to Knock stock or lakeside of Door? So this is 200 tags that we're going to try to get out over the next several years. And we're working again with a variety of partners here, really closely with our tribal partners and the tribal commercial fishers to get fish up in this part of the lake. And it's been going well. We've tagged twice in the fall, so to speak, and then once in the spring, and we've got a couple more bouts of that to go. And so that's all I had for you today. I really appreciate you guys showing up and listening. I'm happy to answer any questions that folks might have.
Okay, I just read your brought thing there. So
Yeah, it can be
thanks very
tough
much,
working out
Dan.
there.
What's that?
I said it can be tough working out there.
Yeah. There was a lot of fun going on in the chat too. Ted Treska caught your joke. So great talk, Dan. Can you, and hopefully people will either just show their camera, raise their hand, or just drop a something in the chat. So tell us about kind of the current status of the fishery. I mean, when it came on like gangbusters, I don't know, 10 or so years ago, I've heard a little bit maybe that things are cooling down a little bit, or that, I mean, what do we know about recruitment or sort of the trajectory of the fishery in the last sort of 5 years?
I know Scott's on here. I don't know if he wants to give a brief update, but if he doesn't, I'll do my best.
Yeah, I can. Sure, no problem. We are seeing declines. We have been for 5 to 10 years. The big year classes are dated back 10, 15 years now. The really big '03, '09, 2015 was the last giant year class. 2015 was huge. It's really sustaining a lot of the fishery right now. But we've had decent ones in between in a relative sense. And by relative, I mean Lake Michigan and Lake Huron.
Right.
So it's holding on, but there are cracks in the armor. It's not to the scale of Lake Michigan. And I'm going to be presenting next week our quotas to our Natural Resources Board and illustrating all of this. So the declines are there. They're being felt. So it's not doing what I think a lot of people assumed it was still doing, but in a relative
Okay.
sense, it's holding on. Yeah.
And
That's
just
like a lot of the things that I was talking about in those first slides was that was like the situation when we started the
yeah.
project, and now the situation is different. There being lots and lots of fish, and at the time probably we were saying recruitment was on the upswing, like when we would have written the proposals and things like that. And then like now we're no longer saying that. Recruitment is occurring is what we're still was saying, which is
Yeah.
like better than a lot of
Right.
places signal wise.
And we've even
Right.
seen in changes in growth, like what you'd expect, maybe density dependent effects. So we're seeing upswings in size at age and condition of fish over the last 5 years, which is reassuring for the status of the population, but might be indicative that of what's going on with numbers out there.
And maybe Dan, one other sort of context question. Um, can you talk historically about like whitefish's use of those tributaries? What's known about that?
Yeah, and again, Scott could help out there. I mean, they did spawn in them. We'll say like, you go back 100 years ago or longer, there were spawning runs, and as many people are familiar on the call, there were also spawning runs in a lot of other rivers, all the way up into the Bays the Knock area and that sort of thing. So there were runs. And I think part of how they sort of got detected is that, people started catching them, right? It wasn't necessarily that folks were out there looking for them. It's just that they started, anglers started catching them. And then I think Scott and the DNR started, doing some sampling, and now they consistently sample them on an annual basis. But it's always really tricky when you sample a spawning run of fish, right? You just think, oh, there's all these fish here. There's
Right.
just hundreds and hundreds of fish. So they must be like producing a lot of offspring and that kind of thing. But, you know, 10,000 fish looks like a lot when if you're up there shocking, but that's not 100,000 fish.
Yeah.
So it's really hard to gauge like how many fish are in the river when you're up there because they're all congregated and
Yeah.
rolling all over when you're shocking.
All right. A couple questions in the chat. First from John Bruner. How does the lake whitefish distribution, I guess like north south, compare to the walleye distribution? I think he means walleye in Green
Yep.
Bay.
So I would say like there's some similar trends in terms of, well, let's put it this way, like the recruitment trends that we're seeing now for whitefish reflect the recruitment trends that we've seen for a longer window of time for walleye, where walleye recruitment in northern Green Bay has been like very low for quite a while. And that's largely attributed to like lack of zooplankton for young fish. So that is pretty similar in terms of that distribution. The recruitment trends are the same, I would
Yeah.
say. Yeah, and the walleye fishery or population in Green Bay is southern Green Bay. Natural recruitment occurs on a fairly regular basis, and it's a very robust, fishery where stocking is not required. And I still think stocking is occurring in northern Green Bay to like kind of bolster that walleye fishery there due
Hmm.
to the loss of recruitment.
Okay. Let's grab Andrew's in the chat and then we'll get you, Nick. So Andrew Hansi said, great talk. Are you in the process or have you considered evaluating percent contributions of river versus reef spawners using in the mixed stock fishery using genetics or other methods, even I don't know, like microchemistry to kind of get at that sort of portfolio effect that you were alluding to?
Yeah. Yeah, one of the reasons that we've continued with the acoustic telemetry as our approach is the new genetics demonstrate that in the Menominee River, there's not a unique genetic signal. So you can't tell like where a northern stock fish came from genetically. Like it could have came from Big Bay to Knock, it could have came from the Menominee River. Southern stock fish could be from reef spawning out in the bay or from the Fox River. So at least currently, the genetic signal doesn't let us get down to that level of precision, if you want to think of it that way, where we could really pick out spawning locations out of a spawning stock. And then the microchemistry, Patrick Forsyth and his group at UW Green Bay has invested quite a bit in that. And similar to like a lot of other studies, it gets really tricky when you have like rivers with similar geology that are located like close to one another. And then you have this big tributary, the Fox, that probably it has a very strong influence on the water chemistry of Southern Green Bay. And I don't exactly know where Patrick is with all of that, but if you got reef spawners that are out in front of the Fox River somewhere, and that Fox River water is like really influencing the water chemistry, it might be hard to really delineate. And so for now, acoustic telemetry seems like the best option for us to be able to say this is a Menominee fish, this is a fox fish, this is a reef spawner. And maybe that'll change in the future.
That makes sense. Okay, Nick.
Hey Dan.
How you doing?
Mike, good. Thanks for the talk. That was a lot of fun to listen to. The my question, just wanted to check my understanding. You said 100,000, like you'll get millions of larvae collected in the bay itself. Is that right?
I don't know that we could say millions, just lots. Like we're just going out and pulling a net, right?
Mm-hmm.
And so like the distribution of fish is really all I could say, like more or less here. We're not extrapolating out to
Mm-hmm.
million. No, no, no.
Okay.
So the millions was adults.
Oh, millions of adults and then hundreds of the thousands.
Yes.
Because that was adults, not larvae.
Yeah, yeah. So the first comment I
Ah,
made
ah,
was adults. So
mhm.
hundreds of thousands of larvae can't get you millions of adults just based on
Yeah,
survival of.
right,
Yep, sorry,
right.
I wasn't following you at first.
Yeah, I was try I that helped me understand it. Thank you.
Okay. I should have
Yeah.
put of adults after millions. I probably made that slide 10 minutes before I got on this thing.
No, all good. Yeah, I was just
Yeah.
I was thinking about the portfolio effect
Right, right.
and right, and I was thinking about the relative contributions of them of river fish and lakes and reef spawning
Yep.
fish, and I was thinking about like my experience with just thinking about tributary spawning fish is when do they go and spawn? They spawn earlier in the year because of temperatures. There's different types of food there. There's a working hypothesis or a working prediction that you might expect the larvae to be larger in size and perhaps have a higher survival. And that's kind of where I was going with it. It's a
It,
difference in numbers, but difference in survival could come in play here.
yeah. And
But
I
I think I
certainly
misunderstood it.
no, yeah. And I certainly don't want people to construe that
Yeah.
we don't value that there's spawning going on in the tributaries because of that portfolio effect. I mean, the fact that we have tributary spawners and reef spawners, I think is a good thing and makes the population like more resilient to potential changes that might occur.
Hmm.
And it's still like we
Of
don't,
course.
if it's millions of fish that are out there, let's say, and the tributaries are responsible for, let's say, 1 million of those fish, that's
Mm.
still really valuable.
Yeah.
But what I think
Mm.
is interesting is more so that maybe it's the reef spawning fish that are really responsible for all these things that we've seen over the last decade, and that
Yeah.
something changed in the bay or changed with whitefish behavior to like make that strategy more productive, and that more fish were surviving to adulthood, and
Yeah.
the
Beautiful thought.
tributary fish were contributing to it. So
Yeah. hmm hmm Yeah. Great. Thanks for sharing that.
So, oh yeah, let's go to Tammy. That was good.
now I'm nervous.
Hey Dan, thanks for the talk. So I guess this kind of dovetails to what Nick was just asking about, but do you think you'll be able to? So one of the things we're seeing in our tributaries is water levels are lower because the bay is lower, flow is lower, and seemingly the whitefish runs seem to be a lot less than they used to be, say, a decade ago. Do you think would the telemetry study be able to show eventually that maybe some of those fish are spawning out offshore on some reefs instead? I'm just thinking maybe it's not as dire as it seems to be. I know when, Scott and Derek and their crew did some shocking this fall. They just didn't see the numbers that they would have a decade ago. So can you speak on that at all?
Yeah, the unfortunate part of the tags last 3 years. So the last time we tagged in the Menominee River was 2020. So those tags are no longer functioning and giving us data. So it would be interesting to have more of like a 10-year tag. And one of the reasons that we use the smaller tags with the lower battery life, one is just they're a lot easier to get into the fish, right? And we're not usually dealing with giant whitefish here. These aren't the Lake Erie versions that, weigh like 7 pounds sometimes. They're generally smaller. But the other thing is we have all these other competing studies, and like the Menominee ends up at times with walleye, whitefish, sturgeon, muskie, all in there at the same time. And so sometimes it's good to have like some fish shut off. But so it'd be really hard to tease that out, like does a high water year mean more lake whitefish might go into the Menominee River to spawn, and then when water is low, those same fish are okay, but they just go out in the bay and spawn, and whether how that would affect contribution, like is the river, even though it may not contribute a lot of fish, is it still like a more effective strategy, or is the reef approach? So that would be really interesting, and it would probably require like those 10-year tags, and then you got to hope water levels kind of go up and down so you get that range for you to test it on.
Thanks.
Yeah, so my answer is no, I can't really tell you anything, but the one thing that we are working on though is the cues and like the timing and how long the fish are in the tribs from those two different cohorts we tagged, and that's the paper that we're currently working on editing. And like, does that differ between males and females, and does that differ between the two genetic stocks?
Are males in there longer?
I gotta remember. I don't think there's much difference
Okay.
between the sexes. I think they might come in like slightly earlier, but
Yeah.
it's not weeks earlier or a week earlier. I don't know if Dan, if he's still on, if he knows that off the top of his head.
Days difference, he says in
There
the
you go.
chat.
Yeah, so
Yeah.
only days difference. Yep.
Hmm.
And the two genetic stocks in the Menominee overlap very heavily. So like in my mind, that means at least in the Menominee, I don't know that. I think you're like hybridization would seem to be between the two stocks is going to be rampant, but
Yeah.
maybe there's some other thing that separates them. I don't know.
Well, it's interesting that Tammy brought up sort of the water level, the habitat aspect too. That's a good question.
The one thing I will say
Yeah.
is we had another grad student that's been working in the Fox River mapping like egg densities of whitefish on the substrate. And we had two very different flow years while he was out there doing
Okay.
that. And there was like changes in where the egg deposition occurred, like the higher density. So they even though water might be lower in the river, there might still be like habitat. It might reduce the amount of available habitat, but there still may be some habitat available to them to deposit their eggs.
Cool. Okay. A couple more questions in the chat. First, Robin DeBruni, are you planning any egg studies in different spawning areas or in the rivers? She's wondering how much spawning is occurring out on the reefs, or if the congregations of larvae are from hydrodynamics, bringing fish from all over together. So have you looked at kind of those physical currents and how that could be? Yeah. Okay.
No. The egg stuff, and I just saw Dale Hansen chime in here. You know, that's something that I know that the Fish and Wildlife Service and I believe UW Green Bay have done some of that in the past, like going out and trying to identify where eggs may be out in the bay. And that was kind of the idea. Dale's a co-conspirator on that one of those projects there where if we can use the grid to like show where the hot spots are, then maybe we can follow up and do the egg searches like in those areas.
Dale, you want to jump in there?
Yeah. Great talk, Dan. Say, I
Thanks.
was wondering, so you were showing the hot spots of where you had detections of fish that were tagged out from the fishery, and they weren't entering the tributaries, right? And I guess the thing that kind of caught my eye was I would have expected that the hot spots would have been very near shore. They're thought to spawn in shallow waters, right? 5 meters or less. But the points were kind of out in the middle. Is that
Yeah, you have to be careful a little bit because of the detection range on the receivers. So like I think some of those receivers were kind of maybe our most inside in that area, and the fish could actually be like farther towards shore than so we can't like pinpoint it. Um, but though some of those shoals are depth wise, I don't know, but they're not necessarily like close to shore.
okay?
Yeah,
Yeah, I mean the little bit of egg work that we've done up on Larson's Reef, the eggs we recovered were from the shallowest points that
Yeah.
we checked, kind of right up on
right.
top of the reef. So
And that could very well be like I can't tell you exactly where they are, right? I'm just trying to help you with they're over here somewhere. Maybe not so much over here, but kind of over here, that kind of thing. And
okay,
again, that's just like our first grab at it. I suspect as we add more fish, we'll probably see some other places like get a little hotter. Um, but
okay.
for the time being, that's what we got to go with.
Well, I mean, I guess it's all speculation, but would you
Yep.
think that they are really, the whitefish are spawning at particular reefs that are out there? Or do you think it's more of a widespread, all along the shoreline, up and down? I mean, they could be using, a specific contour, for example.
Yeah.
And,
I think it's like widespread.
yeah.
I think that's the same situation that occurs like along the lake side of the Door Peninsula. You know, we focus on North and Moonlight Bays, but I think the habitat is suitable all the way up and down that thing. And the one thing I didn't talk about today is, as you know, we also have the whole lake side of the Door Peninsula is now covered with receivers. So like when a fish goes out in the lake, we have much better information on where it is. And those bays to knock fish do really go out in the lake. So and that's why we added them. But I think it's probably like widespread. I mean, even in the Fox River below De Pere Dam, there are certainly like on our heat maps hot red egg densities, but there's like a lot of orange and pink, and we're like it's pretty broad.
Okay, thanks.
Thanks, Dale. So Dan, we've got some questions from Aaron Dunlop asking about some of the larvae. And I'll note, Dan already mentioned Patrick Forsyth, and I think one of his papers was put in the chat by Ted. And we've also asked Patrick to give a talk, and he declined for this fall. He's just taken on more administrative duties, but he did offer to do it in the spring. So we'll try to bring him on as well. But I wonder, Dan, if you're able to answer her question about the ecology of the larvae in the tributaries. Aaron's asking, do they get swept out pretty quickly to the bay? How much time do they spend in the tributaries? And any information on the diets of the larvae that are born in the river versus born in the bay?
Yeah, so that's not something we did a lot of diets and energetics of the larvae that we caught like out in the bay, but we did not catch larvae in the rivers. I mean, we actually attempted it to some degree, but it was very low. And I can't, Scott or Dan, I can't remember if in Andrew's paper they talk about the diets or it's more like the distributions.
Yeah, I don't remember much about diet stuff. It was more
Yeah,
distribution and timing of
right.
larval drift for sure.
But Dan, I'm correct. I don't know if he's still on here. Like
Yep, I'm here.
Jordan did a ton of work with the larvae, and I'm pretty sure we have diet data for those.
Yeah, the diets, we had a really hard time getting sufficient numbers of larvae in the rivers. One of the objectives was some calorimetry work, and we needed a critical mass to burn. We did not achieve that critical mass in the rivers, but from samples collected in southern Green Bay, south of Chambers, northern Green Bay, north of Chambers, and Lakeside, larval diets were all dominated by cyclopoids.
Yeah, and that confirms with sort of historical literature on what they would prefer, but we're seeing differences in their diets now in places where they're struggling, the larvae, but they're not empty either. And Erin's done a lot of work on that too, but they are growing more slowly, especially in
Yeah.
her work around fishing islands. So
Yeah, I don't know if Andrew Ransom did anything either with the timing, like how long do they stay in the river and, before they start showing up in the flume, like out front, which in theory is like either staying in the river if you could keep in the plume coming out of the end of the river where maybe it's a little more nutrient rich would be the advantage to tributary spawning. So yeah, I
yeah,
see Ted chiming in there too. So you got to realize that it ain't most of these, the Menominee, once you get in it, you can almost see the dam. Like it's not a very long run of the river. Acano and Peshtigo actually have a much longer bay to first dam run, and the Fox is a little bit longer, but not real far.
so maybe one other question I'll ask Dan. We were chatting about this a little bit before, and you raised it in the beginning. Sort of one of the ideas for why they started colonizing these tributaries. I've heard a lot of people sort of talk about the spillover effect, maybe, and you even mentioned that too, right? Um, do you think, like, thinking about the whitefish population out in Lake Michigan, probably maybe the largest we've ever seen in like the last century or something, and we didn't detect any spillover effect in the tributaries that was occurring. Do you have a sense of what was just the right recipe that the spillover effect? Is it just proximity to some of these tribs in a smaller place like Green Bay, sort of density dependent still, or have you given any thought to like why there, why now, and how that's and then I think you've also talked about maybe it isn't as important to the fishery as maybe people were thinking or maybe even hoping, but I don't know,
Yeah.
just some speculation on that.
Yeah, I think the proximity, I think at the beginning of this, the spillover was like, oh, well, these must be fish that are swimming from like Big Bay to Knock or coming from out in Lake Michigan, because we weren't really thinking about the reef spawning in the bay. I mean, I think folks have always thought that some level of it occurred, but so I wonder if it wasn't just because like maybe out in front of these rivers or in southern Green Bay that habitat started to get like maybe saturated with adults and they go looking, or maybe it's not even that great a habitat. Like they use it, but it's like I think the fertility that's provided by the Fox River like overcomes some of the issues that we're having with Dreissenid mussels and other parts of the lake, so you have the food. And then in my mind, even like the temperature loggers told us a lot of interesting things. Like the whitefish in southern Green Bay are in like warmer water pretty like more often than we think they would have been, right? But maybe you're willing to do suffer a little bit if you can eat, like feeding
Yeah.
is a little better. So I
Yeah.
don't know. I think proximity matters, but I don't know. Yeah, I don't know why that wouldn't have occurred in some other places. I also think that I don't discount what we've done with the tributaries to make the habitat in those systems better. So that's hard because like maybe the reason they weren't doing it for so long is just because it was just inhospitable
Right.
and the outcome wasn't good. So like you go up there but you don't hatch, and then slowly over time things get better and some larvae start surviving and then those larvae are coming back and then you just build up. I don't know.
Yeah,
Like I struggle with,
you had high site fidelity. That was kind
yeah.
of cool to see.
And that's something that like Scott had seen in previous tagging and the loop tags showed us. But yeah, there are some fish that don't come back to the river every year. They're alive, but they don't come back. Now, if they
Yeah.
do come back, they come back to the river that we tagged them in for the most part. There's a little bit of strain in the Peshtigo and Menominee, but
Yeah.
yeah, I just don't know. I think the key question is, does tributary spawning help you overcome the lack of zooplankton that might be occurring like out on in a reef spawning situation, right? In places where you don't have the Fox River or something like the Fox River providing that fertility.
Right. Which is what I was looking to see. I don't see anybody quickly in a skim from the tribes that are doing some of that like colonization work. I did see Matt, if Matt wanted to jump in
Yeah.
and sort of talk about that potential role of the work they're doing. We've had a webinar about that. Rusty, probably a year ago. I think we probably have it recorded. But Matt, did you want to add anything to this conversation before we go to Ted's question about tributaries?
I mean, we can't answer the question, yeah, we just generally think that at least some of these rivers that have good estuary habitat, you know, might vary from year to year, but they at least have the potential to help, to help provide a lot more food resources than what they're getting coming off the reefs in the main basin.
Yeah. And like I just use Big Bay to Knock as an example. If you don't have spillover, right, from reef spawning, because there's not that many fish out there, I mean, maybe tributaries are like a second choice, right? They'd rather spawn in the lake. I don't know, but like forcing the fish into the tributary, I think is a valid test of whether, I mean, something is better than nothing is the way that I look at it, and it's definitely worth experimenting with. I mean, I'm excited. I've really enjoyed watching like the sort of documentaries and some of the video stuff of doing it. It's a really good idea, and it does look like they have very high fidelity, right? So they'll come back hopefully.
Yeah. Is anybody doing like zooplankton like in the rivers versus we know it's very low on the beaches, like adjacent to reefs. Is anybody also collecting these in the rivers?
We
To kind of
are we've put in proposals in the past to try to do that, and we're actually trying to put together a proposal right now to try and do
yeah,
that. We have not done it, but we definitely want to do that.
that would include some of these Wisconsin trips too, or more the northern part. Yeah,
I'm not
excellent.
sure if Troy Zorn is on, but he was doing some periodic zooplankton collection up in Little and Big Bay in the rivers and then out in the plumes. And yeah, there is a noticeable plume effect. It's low overall, but higher in the plume than elsewhere.
Yeah, thanks, Dan. And maybe, sorry, Ted, I don't want to miss Titus's question here. What is good young of year whitefish habitat in Green Bay? Give that back to Dan. Do you know?
I mean, we just know where we caught them when they were larvae, really. And actually, after that, you start to not really even know where they go, like pretty quickly. That was always something that, where are they when they're like this big? And Scott, they do some juvenile sampling, and I know probably like Tammy, they show up in the they do bottom trawls but I don't know if that necessarily could get you at what's good habitat. Yeah,
Thanks.
I think that's like a big challenge for everyone, right? Is like what happens after the post-larval phase, and even like when they're a little bit bigger larvae, like where are they once they're off the beach?
Cisco and whitefish.
Yeah, where do they go?
Yeah.
Those are hard to get transmitters in, those larvae.
Okay, Ted, thanks for your patience.
Post-tagging mortality is pretty bad, huh?
Yeah.
I was just going to ask, and is there any appetite for doing a VPA type array somewhere, maybe on like Young's Reef in Green Bay, since it seems like you had a lot of hits for those commercial fish around Sturgeon Bay that showed up on, across the bay on Young's Reef. And I know it takes somebody finding the money and the receivers and tagging the fish, but could it answer the questions that Dale and them have been looking at to try and put a VPA on one of those things similar to what they've done on Good Harbor, Michigan DNR?
Yeah, I think that's something that, if Scott, if it has a management application that we can do something with, then we're all in. The thing of it is like, and I didn't show it today, we're kind of working through this strategic plan that we've laid out that I'm sure you've probably seen, Ted, of like we're in these different phases. So those weren't just like nice titles on the slides. They're actually like this laid out strategy of trying to address it. And like next phase, ideally, would be for us to actually go back out on the lakeside and tag again because we're going to have a lot more receivers in Lake Michigan. And we're kind of have moved away from the tribs and in Green Bay and are like back up, we're back up in Big Bay to knock right now and then maybe headed back out to the lake. And then you've got like Nobbin and all these other locations. But I think the way that I operate is if somebody says that this would really like benefit decision making and management, then we would really try to make it happen by writing the grants and that kind of thing. So I think the first thing we got to do is let that study play out and make sure the hotspot stays consistent and it doesn't end up being three or four places and maybe places that Dale has already gone and looked end up being hotspots too. So, that was well-informed sampling, so to speak.
I was just going to say the All Too Clear folks were up on Lake Simcoe doing videos, I guess, this week. And so if you can find a place that might have a pretty consistent, maybe you could convince them to come back and get all the details without as much effort, but who knows?
Yeah, we interacted with them quite a bit when they were like, trying to find whitefish under the ice and that kind of thing at the time they were filming here. So that is another way to do it besides like the pretty laborious approaches that we typically employ. I don't know, Dan might not talk to me anymore if I institute another egg an egg project.
I'm out.
Yeah, you're out.
Just let me know when you're going to do that recreational tagging this winter. Maybe I can make it back.
Next week, I think.
Oh, well,
Yeah,
might be too
I
late.
don't know. We'll see. We'll let you know. Yeah.
Ed Rutherford chimed in just talking about in reference to what's in the tributaries to eat. He was doing some sampling for young salmonids in the 90s and early 2000s. They did find cladocerans, especially below hydropower dams, but otherwise just midges were emerging. So definitely sounds like a data gap to try to nail down how much better that habitat is for feeding. Seems like it should be, but
Yeah, I think the one thing that, you know, like the fox especially, we know it's I think a pretty big producer of walleye and perch and like other species. And let's face it, every single fish needs zooplankton when they first hatch. So unless there's like some mismatch thing going on, there should also be those zooplankton available for whitefish as they come out if it, you know, higher. And I would guess like the Menominee is probably the same. We just know less about, I think, like how many walleye and perch might utilize it and then the output that you get from it.
okay. Well, we had a great turnout. Almost 90 people, I think, showed up for your talk, Dan, which is really cool. Renee has dropped the chat in the reminder for people to sign up for the next two webinars. We're going to try to do some catch up next month. But great presentation. Thanks everybody for sticking around for a great discussion and all the good questions. People can do their virtual applause just to say thank you. And everybody have a great weekend. Appreciate it.